Regeneration methods of plants in culture. Plant regeneration. Let's make its presentation presenting. Plant regeneration, the process of growing an entire plant from a single cell or group of cells. Regeneration is possible because plant cells can be made totipotent using hormones, differentiated tissues, stems, leaves, roots, etc. Undifferentiated embryonic cells are totipotent can become a whole new plant by differentiating into a whole new plant. Explant in nutrient media, callus, embryoid, embryo plantlet. Plant regeneration. The main objective in plant cultures is to regenerate a plant or plant organ from the callus culture. The regeneration of plant or plant organs only taken place by the expression of cellular totipotency of the callus tissues. In agriculture biotechnology, tissue culture is most important for the regeneration of transgenic plants from single transformed cells. Organogenesis. Organogenesis is the formation of organs in either shoot or root. Organogenesis in vitro depends on the balance of auxin and cytokinin and the ability of the tissue to respond to phytohormones during culture. Organogenesis takes place in three phases. In the first phase, the cells become competent. Next, they differentiate. In the third phase, morphogenesis proceeds independently of the exogenesis of the exogenous phytohormone. Indirect organogenesis, formation of organs indirectly via a callus phase, is termed indirect organogenesis. Induction of plants using this technique does not ensure clonal fidelity, but it could be an ideal system for selecting somaclonal variants of desired characters and also for mass multiplication. Induction of plants via a callus phase has been used for the production of transgenic plants in which the callus is transformed and plants regenerated, or the initial explant is transformed and the callus and the shoots are developed from the explant. Direct organogenesis. The production of direct buds or shoots from a tissue with no intervening callus stage is termed direct organogenesis. Plants have been propagated by direct organogenesis for improved multiplication rates, production of transgenic plants, and most importantly for clonal propagation. Typically, indirect organogenesis is more important for transgenic plant production. Let's move ahead. Okay, let it open now. Somatic embryogenesis. Introduction. Somatic embryogenesis is an artificial process in which a plant or embryo is derived from a single somatic cell or group of somatic cells. Somatic embryos are formed from plant cells that are not normally involved in the development of embryos, that is, ordinary plant tissue. Somatic embryoid formation may be formed from vegetative cells of a mature plant, reproductive cells other than zygote or cotyledons, hypocotyle or young plantlets. Hypocotyle cotyledon. The part of the stem of an embryo plant beneath the stalks of the seed leaves or cotyledons and directly above the root. An embryonic leaves in seed-bearing plants, one or more of which are the first leaves to appear from a germinating seed. Steps of somatic embryogenesis. Initiation of embryonic culture, proliferation of embryonic culture, prematuration of somatic embryos, maturation of somatic embryos, plant development. Types of somatic embryogenesis. Two types of somatic embryogenesis: direct somatic embryogenesis, indirect somatic embryogenesis. Direct somatic embryogenesis, the embryos initiate directly from explants in the absence of callus formation. Indirect embryogenesis, here the embryos are developed through cell proliferation, callus formation. The cells from which embryos arise are called as induced embryogenic determined cells. Here, growth regulators with specific cultural conditions are required for initiation of callus and then redetermination of those cells into the embryo development. Advantages: higher propagation rate suitable for suspension culture, artificial seed production, somaclonal variation, germplasm conservation, labor savings. Disadvantages: low frequency embryo production, complete embryo production. Many may create unwanted genetic variation, inability to generate large number of normal free living plantlets. Plantlets are weaker, response 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 respond tissue specific. Factors influencing somatic embryogenesis: auxin, cytokinin, nitrogen, activated charcoal, age of culture. Auxin. In medium having relatively high concentration of auxin, embryonal budding or embryonal clumps have been observed for cell differentiation. The medium should contain auxin and reduced nitrogen. Subsequent development takes place in medium with no auxin or low concentration of auxin and reduced nitrogen. In some plants, first and second stages occur in the first medium, and plantlets development takes place in the second medium. Cytokinin. The role of cytokinin in embryogenesis is not clear. Embryogenesis in carrot cell suspension is stimulated by addition of zeatin in medium lacking auxin but inhibited by the addition of kinetin. Inhibitory effect of exogenous cytokinin may be due to an increase in endogenous cytokinin in growing embryos. Nitrogen. The ratio of nitrogen to auxin is an important factor controlling embryogenesis. Embryo development can be initiated on white medium with low nitrogen content only in absence of auxin. At low nitrogen concentration, organic nitrogen is more suitable than inorganic nitrogen. Substances used as a source of nitrogen are potassium nitrate, ammonium chloride, glutamine, glutamic acid, alanine, urea, etc. Activated charcoal. Presence of activated charcoal in the medium helps embryogenesis in several cases. Activated charcoal may absorb the inhibitory substances present in the medium. Age of the culture. Embryogenesis usually occurs in short term cultures. With older cultures, this ability decreases and ultimately it is completely lost. This may be due to either the inability to synthesize some embryogenetic substances or changes in the ploidy level, which may lead to loss of morphogenetic potential. Thank you.